late as not this.
I saw you. Oh, you made it there?
Yeah.
I watched a little bit.
I played left-handed though.
Oh,
Yeah.
a little handicap?
Yeah.
It's so
I
good.
tore my labrum, not playing pickleball, just
What?
at the same time as I was playing that. And but I didn't want to like not show up. So I played left-handed. No, I'm terrible. Actually, I think I was better left-handed. My teammate was carrying the team. Yeah, exactly. Okay. Is there usually a mic in here, or does
No.
Okay. I don't have a very loud voice, but I'll try my best. If you can't hear me, I'll try to project more. Can you hear me right now in the back? Kind of.
We'll be quiet.
A little bit. All right. Well, feel free to move up too if you can't hear enough. I'll try and project though. Yeah. So Dr. Troy's out today, so I'm just kind of filling in. But it's all her slides. She's kind of given me verbatim kind of words to say to you. So might seem a bit scripted at times, but then after I get through that, I'll kind of go awry and just do a little bit of a discussion with you. So make sure that you understand it all. I think. So what was your last lecture? Oh, okay. So blank slate. Nice.
Yes.
Awesome.
Start of a new unit.
All right. So I know you're following this up with cranial nerves. So some of this will pertain to that. You'll be having, your spinal cord lecture as well. So a lot of this is kind of setting the stage and seeing how these pieces will connect back. But anywho, stop me along the way if you have any questions. Again, for those that know me, don't know me, Dr. Urbanik. Teach anatomy kind of across the board. So PA, anatomy, undergrad, 3135, 3136, some dental stuff. You'll see me floating around. If you ever have questions and you can't get to Dr. Troy, feel free to stop by. I'm somewhere on the 4th floor. But it's good to see a lot of familiar faces. Okay, so if we are kind of talking about the brainstem here. So when we're talking about the brainstem, we can divide it into 3 kind of major functions. Right? The first of which is conduit function. So conduit basically providing a route of transmission from one place to another. Essentially what that means, right? So conduit functions for the brainstem, right, are going to allow all those ascending fibers to make their way to the thalamus, right, or the cerebellum, right, as they come up from the spinal cord. And on the flip side, all of your descending pathways, right? So all those descending fibers that need to reach the spinal cord, right? And all of the kind of numerous, relay nuclei in between, right? So that's one major function of the brainstem, right? Passage of ascending and descending fibers between the places that they need to go. The second, I added these cool trans effects, right? Like that, it pops up, so it's not all at once. The second is cranial nerve function, right? So you're going to have a whole lecture on this coming up soon, but all those cranial nuclei are going to be present within the brainstem, right? So with the exception of olfaction and optic, right, which are tracks, so direct projections. You probably remember this from 3135 to some degree, right? To the cortex. All the other ones will have a nucleus associated with it, right? So when you're looking at those, they're going to project or emerge from the brainstem, right? And typically ventral, right? There's only one that goes on the dorsal aspect. Do you remember what that was? Trochlear, right? Cranial nerve 4, the only dorsal emerging cranial nucleus. We'll talk about that again, right? But as they emerge, they'll make their way kind of anterior or ventrally, right? Pass through a foramen of the skull and make their way to the tissue that they're going to supply ultimately. Right? So we'll get back into that. But cranial nerve function, right? They're housing those nuclei for 10 of the 12 cranial nerves. And thirdly, we have integrative function. This is a bit more complex. Yeah.
What were the 2 that were like within?
Yeah, so olfactory and optic. So those aren't going to have a cranial nucleus associated with them because they're actually direct projections from the cortex. So this would be cranial nerves 1 and 2, but you'll have a whole lecture on that, so don't get too caught up on that yet. But the integrative functions, right, within the brainstem, these are going to be kind of complex motor, systems or motor complexes, and they are going to have, integration of, inputs from different regions because they're going to control, it's going to control complex functions like respiratory mechanics and cardiovascular reflexes, spinal reflexes, things of that nature. So it's taking in inputs from many areas and has outputs associated with many areas. So highly complex, highly integrated. We'll talk much more about that. And specifically, a lot of these integrative functions are going to occur in what we call the reticular formation. So it's an important piece to write down, but we'll talk a lot about that in a few slides. All right, so the reticular formation you can think of as the core of the brainstem, right? Core of the brainstem, specifically the core too of all those integrative and complex functions of the brainstem. Any questions on that? Okay. So, a little bit of anatomy, right? Just structures. You've probably seen a lot of this before. Also, there's a really helpful, actually, ironically, I think I first learned of it when I was a student in this class, taking it with Dr. Choi, but neuroanatomy.ca, has she given you that yet? Yeah, so that's really helpful when looking at these structures. It goes through all the slides, coronal, sagittal slicing, gross images, toggle labels on and off. So diagrams are great like this, but they're static, so you can't really move through them, right? Granted, the images are static, but you can go slice by slice and play around with that, which is helpful. So take a peek at those as well. But this is just straight out of the Blumenfeld text. We're isolating essentially the diencephalon and the brainstem, right? So when we're looking at this view, if you haven't seen this before, this is showing you kind of a scout on an image highlighted what we've kind of dissected out and zoomed in on. But we have the thalamus up here, right? We have our pons, right? The midbrain, the section essentially connecting the two. And then inferior to the pons, we have the medulla as it becomes continuous with the spinal cord. So that's what we're looking at here, just generally speaking. But when you talk about the brainstem, as you may have heard, it's made up of 3 components. Do you know what they are? What do you think? Yell them out. Yeah.
It's the midbrain, the pons, and the medulla.
Yeah, correct. 10 points. So midbrain, pons, medulla, right? Basically from rostral to caudal, working your way down. That's exactly correct, right? And when we look at the medulla, right, which is this region here, there are a number of structures I wanted to point out. So when we're looking, kind of first and foremost, in midline here, we have our pyramids, the pyramids of decussation. So we'll talk about those corticospinal fibers, right, that are going to be descending through here, majority of which cross, but a small population do not, right? They remain ipsilateral. But we'll talk about that in a moment. And then if we move lateral to that aspect, we have our pyramids. Our olives. So the olives are going to be just lateral to the pyramids here. They'll contain something called, bless you, the inferior olivary kind of complex or nuclei, which will be important in a moment. And then in between kind of the sulcus, between pyramids and olive, we have some cranial nerves emerging, right? And just lateral to the olive, we have a number of cranial nerves emerging on that surface as well. So you can see how they come out, right? Now, if we were to kind of look at this posterior view, so we've taken this, rotated around, we can again see the structure of the medulla. But the medulla is going to have this 4th ventricle associated with it. And if you follow it down, you see this inferior apex here, where it's coming together, it's narrowing. Right? Remember the 4th ventricle narrows into the central canal because ventricles in the CNS are continuous and the spinal cord is part of that. So the central canal runs into the spinal cord, right? So that's where it's narrowing. But the point at which it narrows we call the obex, O-B-E-X, obex. All right. And then, a little bit on this anterior view. When we're looking at these pyramids, if you were to do sections through them, and again, you're going to get into this with your spinal cord lecture, but you can start to see the crossing of those corticospinal fibers. So, they are going to start on the ipsilateral cortex, right, make their way down through the internal capsule, and they come out right into through the pons, they make their way into the medulla, at the pyramid. 85% of them cross to control the contralateral limbs, right? We call that the lateral corticospinal tract. 15% or so remain ipsilateral, right? So they do not cross. We'll do that in a later aspect. But those are the 2 tracks that are forming there, just for reference. Okay. When we're looking at this posterior view of the colon, because it's kind of hard to see, sadly, but when you're looking here where the spinal cord is becoming continuous with the medulla, you can see that there are these bulbous sections at kind of the top. There are these columns. Do you remember the posterior columns or the dorsal columns? Right, that pathway that's bringing all of that kind of general sensory information back up. Remember, conscious proprioception, touch, pressure, vibration, things like that. Right, so as they ascend, right, stuff's going to add on, right, starting all the way down the distal aspect of the spinal cord, and if it's below T6, right, it creates that, adding on to that fasciculus or group of axons in the CNS called the fasciculus gracilis. I always like to think gracilis is a muscle in the lower extremity, so gracilis is for below T6. And then after that point, all that sensory information coming in begins to form another fasciculus lateral to it. That was the fasciculus cuneatus, sensory information from kind of the trunk and the upper limbs, right? As that comes onto the spinal cord. But once the spinal cord makes its way into the medulla, right, those are going to begin to synapse in nuclei. Sometimes they're called collectively the posterior column nuclei, but they have a name. It's the nucleus gracilis and the nucleus cuneatus, right? Or the gracil nucleus and the cuneate nucleus, if you want to say it that way. Either or is acceptable, right? So those are those bulbous structures you're seeing here. I guess that was my long-winded way of saying you see these columns coming up, the fasciculi. And in midline, right, the fasciculus gracilis is going to make its way into this bulbous structure in midline, just below the ovax, called the nucleus gracilis, where it synapses, right? Lateral to that, the fasciculus cuneatus, as it ascends, makes its way into that bulbous structure called the nucleus cuneatus, and it synapses. And then if you recall from there, they cross and run as the medial lemniscus. Does that bring it back a little bit of memory? All right, second. Okay. So another structure that you mentioned earlier was the pons, right? So the pons is going to be kind of this ventral, kind of middle aspect of the brainstem. You know, as you rotate around, you'll see some better views of it. But it's right here, and it literally means bridge. So the Latin word for pons means bridge, and it's just that. It's a bridge between the cerebellum, right, and the pons. So it connects the two cerebellar hemispheres. Although they're not, you know, simple direct connections. You have these widespread groups of nuclei, right, in the cerebellum, and each of them kind of has this, you know, kind of more complex indirect pathway between the cerebellar hemispheres and the pons, and it does so through these peduncles. Remember, the peduncles are, if we look at the posterior here, these sections, collections of axons. They've been cut here, but this was the middle cerebellar peduncle. This was the superior cerebellar peduncle. This is the inferior cerebellar peduncle. Kind of grossly, you can think of it as, you know, those bridges holding the cerebellum up into the brainstem. You'll talk more about what travels in each of those, but typically the inferior cerebellar peduncle is going to be all that information coming up from the spinal cord going directly into the cerebellum. So it doesn't really pertain to the pons. But the middle and the superior will have connections with the pons and pontine nuclei, whether it's directly or indirectly. So middle cerebellar peduncle is more of a crosstalk just between pons and cerebellum, and then superior is going to be more of an output that's flowing up back to the cortex, or basically. But that's kind of generally how it's laid out there.
Before you talk about the 85 and 15%,
Yeah.
85 contralateral, 15% infralateral,
Mhm.
is that motor or sensory?
It's motor, yeah. So it's descending motor information. Anterior corticospinal typically is going to control like axial musculature, so things more for posture and balance. But again, you'll talk about that on its own lecture in detail. So, but that's a good starting point.
Thank you.
Yeah. Okay. So this is more of a gross specimen of actual tissue clearly. This is a sagittal section, so midline view. And we're looking really isolated at the brainstem and the cerebellum, right? There's a little bit of diencephalon in view. So just for orientation, right, this would be anterior, this would be posterior, right? Superior, inferior. So we see our pons, right? We see our medulla as it becomes continuous with spinal converge from what we talked about here. So if we're following this inferiorly, the opex is going to be right where this begins to taper into the central canal, so right down in here. But I really wanted to show you this for the kind of region of the midbrain. So the midbrain, if you were to draw kind of two lines, is going to run here to here, right? Just excluding the pons there. So this whole section is your midbrain. Right, in the midbrain, you'll kind of notice, well, not the best view for it, but I'm sure you've seen it in another view. You have these four bumps, right? These four kind of bolus projections here. So you would see two of them because this is a midline. There's a superior and an inferior one. They call this the superior colliculus and the inferior colliculus. But there's one on the right and the left. So there are actually four in total. So two superior colliculi, two inferior colliculi. And if you wanted to use kind of the Latin term, the corpora quadrigemina, which literally translates to the four bodies, right? So that's what it's talking about. The two superior and the two inferior colliculi kind of on that posterior aspect of the midbrain. They're going to be really important. We'll talk about that down the road in a second. Actually, I think we'll talk about it now. So, there's a mnemonic I like to use, and it's not in here. Well, I guess you're not seeing what I'm saying all the time. But eyes over here, so you ever heard that? So, superior colliculi, visual reflexes, inferior colliculi, auditory reflexes. So, eyes over here. So, it's a great way to kind of remember them functionally. And then you can place in all your little kind of connections and pathways to that for the detail. So it's a great way to start. But the superior colliculi are going to be really important for directing visual attention, coordinating eye movements, reflexes, right, as it pertains to visual stimulus. And the superior colliculus is going to then project to a lot of the brainstem areas that are going to be involved with those functions. So like the association cortex, the visual kind of cortex, visual area, right? Visual motor area, they call that when you learn about the cortex and Brodmann's areas. And it's also going to relay structures like the pulvinar, the very kind of posterior aspect of the thalamus, right? And also right, the lateral kind of posterior nucleus, lateral genicular nucleus, right? Connections to the visual relay centers, right? So let's take another look at that, back to this view. So if we're looking at posterior view, midline, right? Here are the colliculi, right? Here are the two superior colliculi, here are the two inferior colliculi, right? And if you look at them, right, you see these extensions laterally, these projections, right? Almost like an arm. Call it a brachium, right? Brachium literally means arm. So the brachium of the superior colliculi, right, this arm or extension, so these are all tracks running through there, right, are going to be a collection of fibers, right, that are going towards things like the visual cortex, optic tract, right, but a lot of these fibers will bypass the thalamus. So it's called the extrageniculate pathway because it's outside or it's not going to the thalamus, right, the lateral geniculate nucleus, extrageniculate. So they're really important for kind of some of the visual reflexes that won't involve kind of the cortical input, which we'll talk more about down the road. Okay, so if we're looking inferior, we have our inferior colliculi here, right? The brachium of the inferior colliculus is going to be a continuation of our ascending auditory pathway. Right, so it's going to project from the inferior colliculus to the thalamic relay nucleus for hearing, or audition, which is what? Remember that? A little bit? Maybe?
What?
What's the auditory relay in the thalamus? Yeah, mediogeniculate. Nice. So the inferior colliculus you can think of as your main midbrain nucleus for auditory pathway. So in addition to its outputs, it's going to receive a number of inputs. So inputs for your inferior colliculi will be coming from several different regions of the auditory pathway and the auditory cortex. So think superior temporal gyrus, the Brodmann's areas associated with like Urbanikis and Broca's, that whole cortical region. So it's pretty diffuse. It's going to integrate a lot of auditory, cortical inputs. Okay. So if we're looking back at this view here, right, we kind of zoom in at the brainstem, kind of rotate it out. So this is now an anterior view. So this is the pons, this is the medulla, here's the midbrain. Right? So it's kind of taking this structure, just rotating it 90 degrees, anterior facing you. So the ventral surface of our midbrain is called the tegmentum. And it's formed by this here, and if you were to look at that in isolation, it's the cerebral peduncles. They're coming down. So the tegmentum is formed in part by the cerebral peduncles. In between them, the interpeduncular fossa, right? That shallow kind of concavity or hollow, literally means hollow, right between the peduncles. So that's the tegmentum or floor, right, to the midbrain. So something that you'll note right in that tegmental area, kind of within the interpeduncular fossa, are two cranial layers that emerge right at the superior aspect of the pons. Do you remember what these are? They're also labeled, so kind of it's a giveaway, but what do you think? Oculomotor, right? So oculomotor, cranial nerve 3, right, is going to emerge superior aspect of the pons within the interpeduncular fossa. So it's one thing you can use as a reference point. So peduncles, if you recall, right, it's going to be made up of ascending and descending pathways that are going to be linking the cerebrum back to the brainstem, or vice versa, right? So all of that information that was coming from, essentially the peripheral tissues, upper extremity, lower extremity, trunk, all of that information coming up, sensation-wise, or all of the motor information going to it, right? They're making their way through the cerebral peduncles. It's kind of the bridge or highway, so to speak, between the cortex and the brainstem. So a number of fibers traveling within the cerebral peduncles. And so part of the midbrain making up the tegment. Okay. But another structure that we'll see that is embedded within this is the reticular formation. And we'll have a nice section view to show you how the reticular formation kind of flows between these structures. So it's going to be present in the midbrain, more so within the tegmentum as well. Okay. So, also, right, we said the brainstem is going to contain all of these ascending and descending pathways, right? And again, in here it says, don't go too far into it because you'll have a lecture on it. But there are some major descending pathways and some major ascending pathways, one of which we've noted already, right, corticospinal. But when we're looking at these two cuts, this is showing you, right, a section of wood back here. We took a coronal section right through kind of the rostral aspect or the caudal aspect. You can see that there's some changes in terms of the position of these tracks as they ascend or they descend. And when you're looking at this red section in the caudal medulla, closer to the spinal cord, right, we can see that the corticospinal tract that we noted is very far ventral, right, as it's entering into the pyramids. And then at the spinomedullary junctions, if we go back to this image, right, so where the medulla meets the spinal cord, so spinomedullary junction, right, at that point, right, most of the fibers in the pyramids have now decussated or crossed to the contralateral side, and that's where we talked about earlier, about 85% of them crossed. And then as you continue into the spinal cord, those 15 remain the same. And that's lateral corticospinal versus anterior corticospinal, but ultimately all destined, right, for motor control. Descending efferents. If we're looking at the ascending, right, and we could, I don't want to get too into the weeds, but I can pull up other slides that show you these tracks more in detail, but I think you'll get into it later, so I don't want to go in there. But for the ascending pathways, the two major ones we're going to be talking about are spinothalamic. Right, spinothalamic is going to be conveying sensations of pain, temperature, and crude touch, sometimes called like noxious, where tissue's actually being damaged kind of touch. Right, So that's all gonna be conveyed by spinothalamic, right? And that's going to run laterally here, also sometimes called the lateral spinothalamic tract, right? So that's gonna be this green section here. And as you work your way rostral, right, you can see that they're still positioned relatively at the same point through the medulla, okay? The other pathway that you're gonna be talking about a lot, and I know I just mentioned it earlier, those are gonna be the posterior column pathway, sometimes called the dorsal column pathway, right? So put it all together with, sometimes it's dorsal column dash medial meniscus. They all mean the same thing. So just stick with the terms that you are given next lecture, your spinal cord lecture, but it all means the same thing. It's just referring to the columns that are forming on either the dorsal or posterior aspect of the spinal cord, whichever term you want to use for position. So again, that was going to be conveying unconscious, right? Conscious proprioception. You have your touch, vibration, pressure, right? So that's that's what's all being conveyed. And that's going to be ascending, as we said, into fasciculi. Gracilis most medial, cuneatus most lateral. The gracilis is for kind of the lower extremity, so foot, leg, thigh, lower trunk. Right? Sometimes you use the distinction below T6. And the cuneatus, right, or upper extremity, hand, forearm, arm, shoulder, right, upper trunk. Right, that's all going to be above T6. Right, so those are the two fasciculi you're following in the spinal cord. But when you get to the medulla, right, you're going to see that those are going to, right, make up our posterior columns. So midline, we said was gracilis. Out laterally was cuneatus. But at the point of the medulla, we hit the nuclei, which collectively is called the posterior column nuclei. Most midline is the gracil nucleus, lateral cuneate nucleus, right, where those fibers synapse on the next neuron in the chain, right, then they cross. And you can see the crossing would happen as we make our way rostral. And once we cross, we then ascend in the midline, what we call the medial meniscus, right, and that's going to go back to the thalamus for relay. Okay, any questions on that? Yeah.
What was the type of information that was called medial meniscus?
Yeah, so it's going to be conscious proprioception, touch, pressure, vibration. She doesn't have it in here, but I know you'll go into them next one, but it'll have the dorsal spinal cerebellum, which is unconscious proprioception. You'll probably talk for descending, you'll do more like anterior cortical spinal, prospinal, the reticulospinal. So you'll go into a lot more of them. And a lot of you probably remember from 30 and 35 the major ones, so you'll hit those again. Probably add a couple more in. Granted, that's down the road, and I don't see what she's presenting yet. So. Okay, so reticular formation is something we kind of alluded to. It's a very interesting structure. You can see this line in purple and green kind of descending down the medial or midline aspect of the pons and the medulla and down into the spinal cord. So it's rather diffuse, but though it's kind of said to be unorganized, it's really not. It's just because it's integrating many different inputs and many different outputs, right? So it's highly integrated with a number of structures, and that's why it has this more diffuse kind of nature to it. But the reticular formation, as we said earlier, this is your integrative kind of center of the brainstem, right? So major player there, kind of critical, the core of the brainstem, so to speak. Right, so highly convergent, highly divergent. So one input can diverge to many outputs, or vice versa. Right, many inputs to one output. So many complex functions are going to be accomplished through the integration of your reticular formation, essentially every other region of the brain, right, in spinal cord. So, specifically, let's talk about this a little bit. Kind of run through these zones for you. So when we're looking at the reticular formation, which in this, I know this is small on my screen, but I think you can see it fine. So when you're following in purple, right, as we're coursing through the midbrain, in green through the pons, green through the medulla, green into the spinal cord, and each of these scout lines is showing you what that section looks like if you took a transverse section. Does that all make sense so far? Okay. So when you're looking at this, right, this is most superior or rostral, this is most caudal once you're in the spinal cord. And you can divide, it's gonna look a little different where you are, but you can divide them into the more lateral zones and the more medial zones, right? So when you're talking about the lateral zone, right, the lateral zone kind of is gonna be found more so in the rostral medulla and the caudal pons. So if we're looking at the pons here, we're looking at the pons right here, we're looking at the medulla right here. We're talking about these zones out laterally. And the lateral zones are going to be really important for integrating the cranial nerve input, cranial nerve function, specifically as it pertains to kind of reflexes and visceral function. Right, which makes sense because your cranial nuclei are going to be in those regions, right, as you kind of move from rostral to caudal. So they're taking inputs from all that, integrating. Medial zones, so as we make our way towards the midline, right, so in kind of this zone, not quite in midline, but more medial along these paths, right, so in here, right, those medial zones are going to be kind of straddling what we call the raphe nuclei, which is kind of most midline. We'll talk about that in a second. But alongside the raphe nuclei, the medial zone, they're going to be responsible for kind of all of the longer running, right, ascending and descending projections. Right, so you're going to see that the reticular formation has tracts that develop within it. So let's talk about those. These are where those tracts begin. So these projections are really important for the control of movement, more so think like reflexive movement or kind of the modulating of movements. So if we're looking at the reticular formation, there are in the pons, in this medial zone, these fibers are going to be making up what we call pontine reticulospinal tracts. I think you'll go more into this, but pontine reticulospinal. And if we move down into the medulla, right, you have your medullary reticulospinal tracts being formed, right? These are the medial zones of those two regions. So these tracts that are being formed, these long ascending or descending projections from these nuclei, these cells, right, are going to be important for, right, kind of altering movement, coordinating movement as it pertains to, say, like a spinal reflex arc, right? So things that happen quickly and without that kind of conscious input. So it can modulate that. It can also modulate kind of ascending pathways like pain or sensations. It can alter kind of perceptions of things. So they're highly integrated. So there's an example in here to talk about. So a cat whose brainstem has been surgically or spontaneously removed, right? From the dying, basically everything is gone, just brainstem is intact, diencephalon down. And we're looking at that cat, right? And these were actually performed, right? Probably back in the day. But that cat is going to still be able to walk, run, right itself, right? It can still assume postures, right? Without any of that kind of cortical input and all of the motor movements you would think of. Where do those, neurons originate? You think of just typically, I'm going to step. I'm going to move my right leg. Where's that first order neuron in the brain? Like primary motor cortex. It's a gyrus before the central sulcus.
Precentral.
Yeah, right. So, right, if you would think if you remove that, how could you move, right? That's kind of what this study was getting at. But they could still walk, they could still run, they could still yawn, right? All these crazy things. In humans, right, when infants were born without cerebral hemispheres, right, they too can still suckle and yawn and kind of do some behavioral and kind of oral movements, right? So they too don't have any of that cerebral cortex. And that's because of the reticular formation, right, that has the ability, right, to coordinate movements, right? It will also integrate with cortical function, but it can still do so without it. Right? So it's pretty wild stuff. All right, so the other kind of aspect we just mentioned is the raphe nuclei. It's kind of that third longitudinal kind of pathway in the brainstem. Raphe nuclei literally means seam, like S-E-A-M in Greek. So it's like the midline seam. So it's going to be running most medial or midline, so to speak, of those three. So there's the lateral zone, the medial zone, the reticular formation, and in midline, the seam, right, is the raphe nuclei, descending down that path. And the raphe neurons are going to project widely to almost every part of the central nervous system, and they play a major role in attention and arousal. So attention and arousal are a major function of the raphe nuclei. And something that's really important that it does, in addition to that, is that it can modulate pain. It can modulate our sensation and kind of interpretation of pain. And that's what we're kind of looking at in this next piece here. So to orient ourselves, we have, this is kind of a cartoon schematic of neurons interacting with one another, and I'll kind of tell you what each number means as we run through this. And this is showing you kind of that same pathway, or on a neuroanatomical view. So we have the spinal cord, up into the medulla, the pons, up into the midbrain, and then we can say where were things going to, where are inputs coming from. The blue is showing you kind of the ascending or afferent, right? Green is showing you some of the inputs coming down, and the red is also descending. So just to orient ourselves a little bit, but I'll walk through this. So when we're looking at pain, right? Because before we even get into the pathway, so we said that the reticular formation, right, has this major kind of modulation of pain component to it, right? So it can suppress it, and it can also exacerbate it, right? So it can go either way. And if we're looking at pain, we said it's gonna, and again, you'll get more of this, but we said this is traveling in what path? What pathway? Remember, pain sensations.
Spinal fluid.
Yeah, spinal tract. So it's coming in, and it crosses immediately in the spinal cord, and then it ascends, right? And as it ascends, it kind of passes its way through the brainstem, the pons, makes its way to the medulla, where some information is going to the thalamus for relay, but some is going to actually go to what we call the periaqueductal gray. The name kind of implies where it is. Peri means around or surrounding. Gray is referring to the colors where cells are going to be gray. White would be violet, so axons, right? And it's surrounding the aqueduct. So it's this group of cells that surround the aqueduct. Periaqueductal gray. Aptly named. Some things in anatomy are. But they're coming in here, and information about pain is going to make its way to these cell bodies here. So that's, this blue line is number 1 over here. These cells kind of number 4 technically. But when this comes in, right, this is telling you, you know, you have pain, right? You cut your finger, you stepped on a nail, right? This information is coming up. And then the hypothalamus and the amygdala and many of the cortical regions all input to these neurons. So depending on kind of your state, right, your mental state, your mood, you know, what your task is at the time, you know, whatever, everything that's happening around in your environment is going to kind of play into this complex integrative function, right? And those neurons could maybe say, we're going to increase pain, or we're going to decrease pain, we're going to modulate it one way or another, right? So it's highly dependent on the individual basis here. So again, number 1, so these are these kind of the pain information coming in. So from the pain receptors, the spinal thalamic tract makes its way in, and then our other kind of cortical inputs are coming down from, like I said, hypothalamus, the amygdala, cortical regions, and then the periaqueductal rays outputs, right, are going to project to a number of regions, adjacent regions. One of them is the raphe nuclei that we mentioned, so that midline kind of structure, and that long descending midline structure. And you can see that it can then come back down to the periphery and modulate. So essentially you can suppress the transmission of pain by this, like I said. So an example of this would be, you know, someone that's in, say, combat, right, and they're shot, right? And maybe they, in shouldn't be able to continue to move, right? But they have, a lot of, say, emotion or fear maybe, or drive coming in from the amygdala. They have the whole kind of executive function and task that they've been set to do coming down, and they can kind of override that pain and just, maybe not eliminate it per se, but they can dampen it, suppress its, potency, and they can continue to do whatever they were tasked with doing, right, through this pathway. So that's kind of one classic example people will use. If you wanted to take more of a research approach, right, if you electrically stimulate the periaqueductal cells there, you can actually eliminate chronic pain in patient populations with limited side effects, right? There's studies where it's so effective you can actually perform operations on patients by stimulating these neurons. So it acts as an analgesic. And it's also how opiates work, right? Morphine that's typically given, right? That's going to bind to these receptors, right? To activate this pathway, right? In order to suppress pain. Right? So it's kind of a very important pathway. Yeah, that is everything she had. So what about questions? Things that were a little gray. Things that you want. Very questions you had. Wanted me to go back to anything? Structures. Yeah.
On the last slide, what was 2 and 3, did you say?
Yeah, so when you're looking at this slide, we give my little highlighter out here again. So when you're looking at this, 2 and 3, these are kind of inputs that are coming in from different. So let's see, I should have set this up. I'll give you the exact labeling because there's like a little blur for the figure itself. So, it's figure 1121 if that helps in the text, but I'm looking at it for you right now. So 4, these are the projection neurons from the periaqueductal area. So these are the neurons in the periaqueductal gray. So they'll correspond to this green right here. 2 and 3 are going to be inputs that were coming in from areas kind of in this region. So you don't necessarily need to specify which, it's just showing you those 2. And then 1 is showing you the inputs that were coming up from the spinal thalamic tract. And then 5 was raphe nuclei, right? That longitudinal pathway that was stimulated through that. And then this is showing you back down to actual inhibition or silencing, so to speak, of pain or somatosensory pathways at the spinal cord. What about structures when we're looking at imaging? Any questions you had on kind of the gross specimen? Do you still have a lab component?
Yeah.
So you'll probably have a lab associated with this, or see them again in hand. Okay. Any of that you wanted to run over? Okay. If you don't have any questions, you don't have to. Just if there's anything that dots that got connected somewhere along the way, we have to discuss it.
Briefly go back over the superior and inferior colliculi of the brain.
Yeah. So we're looking over here. So we're kind of start inferior, right? So inferior colliculi, inferior collicula singular, right? That's going to be essentially the continuation of that auditory pathway that was coming up, right? So continuation of the auditory pathway, and it's going to be really important for projecting to, right, the auditory kind of relay in the thalamus, the medial genicular nucleus. So that's going to be where it's positioned. So auditory kind of pathways, reflex, or relay to the thalamus. It's kind of its brachial arm. Yeah.
So the branchium of inferior colliculus, that doesn't go to the thalamus, or that's a pons?
So the brachium is basically just like that rounded structure that we're seeing made up of the tracks, you know, of all those accents passing out of it. So if you were looking, if she had like a diagram, I could even pull up a diagram that might be, show you like the actual like point-to-point connections, if that would be easier. See what I can find real fast. Well, I'll just talk about it instead. It's just basically auditory information is coming in, synapses, passes to medial geniculate nucleus. So it's just like relaying to, and then that's going to relay up to the cortical, you know, auditory areas. So your primary auditory cortex, and then you can have your tonotopic mapping, right? So on and so forth. You can kind of follow that up. I mean, there are reflexes as well. It's not in what I was given, so I'm going too far down that pathway, but you know, like the superior colliculus, for example, right? There are some fibers that you'll see that will pass, you know, to the thalamus. There are some that in the extragenicular pathway will not, and then they're going to coordinate kind of reflexes, like visual reflexes. It's the same for auditory. They kind of, you know, control your auditory reflexes that are going to kind of bypass those routes, if that's helpful. So think like visual and auditory reflexes in addition to taking the route you would typically think of.
And the inferior hearing MGM, superior seeing LGM.
Perfect.
Okay, thank you.
Yep. Because if you think of like the visual pathway, right, you have the retina, right? And then from the retina, you have your nasal and temporal. They're going to come down together. And then you get a crossing, right? So you have the crossing of fibers at the chiasm. Then you have the optic, what do you think, tract? You guys remember this a little bit? So optic tract, and then that's going to come back to the medial genicular nucleus. That's going to relay via the optic radiation. You have Meyer's loop, right? That's going back to visual cortex, right? Kelpic fissure, Brodmann 17, so to speak, and you're looking at that, right? That's the typical pathway. But also, when you look at those tracks, or the chiasm, depending on where you're looking at, there's information that goes to the colliculi. And then the colliculi takes that information and can perform some reflexes by then going back into the brainstem through some of the pieces we were just talking about to perform a reflex or, you know, whatever it may be that you're looking at that's bypassing that other pathway. So that would be something like, you look at, you know, pupillary light reflex, the accommodation reflex, you're doing like horizontal gaze, vertical gaze, that stuff that's going to integrate a lot of that function coming from superior colliculus. But it does interplay. So I'm guessing she's going into all of that. I think this is more of just understanding the structures at play, and then you're going to look at those pathways, because there aren't any pathways in the slides. But yeah, that's a great question. Well, if you don't have any questions, I guess have a fun and safe National Marquette Day.
Oh yeah. Yeah. I'm gonna see you out there.